Today, it's a privilege to introduce our speaker, Saurja DasGupta. He is an assistant professor in the Department of Chemistry and Biochemistry here at Notre Dame. He studies RNA structure, function, and evolution, and how they relate to each other. His lab has an ambitious goal, which is to understand how life emerged on Earth. So maybe he'll tell us more about that today. He holds a bachelor's degree from the University of Calcutta and a master's degree from the Indian Institute of Technology in Bombay. He earned his doctorate in chemistry from the University of Chicago. And after that, he was a postdoc research associate at Massachusetts General Hospital, Harvard Medical School. Today, he will talk about where we came from and how we can return to our earliest beginnings. He also has some posters here on both sides of the auditorium in comic book form, which illustrate important scientific concepts related to research into the origins of life. And I encourage you to come up and view them after his talk. Welcome, Dr. DasGupta.
Thanks, Dina. Okay, can you all hear me at the back? Okay, wonderful. Thank you, Dina, and really thanks a lot for coming here, stopping by for some quick science in a football weekend. We grow up and we go in different directions, we end up doing different things, but we might end up doing different things, but when we're born, we're all born scientists, and we have asked these crazy questions that we never really found good answers to, right? I'm sure you wondered how vast the universe was. You've wondered what consciousness really is. But because we didn't really find good answers to them, we moved on, then we grew up, and then we replaced those questions with maybe more existential questions like, should we cook or order Grubhub or go out, or should we watch Netflix or Hulu or Amazon Prime or all the other options that we have nowadays? But today, what I want to do is bring you back to when you're a kid and remind you of one such question that you must have asked at some point in your life, which is, how did life begin? Where did we all come from? Okay, so this question of the origins of life is very many different things to different people, but it is a question that is at least as old as human civilization, and it's 4 billion years in the making. But we have to understand that the origins of life is a historical question, meaning that we may never really outline the exact chemical and biochemical steps that it took for life to emerge, from a primordial goo 4 billion years ago. But that does not mean that all hope is lost. So the origins of life, and this is something that I want you to take back with you, is it's also a scientific question. And today I'm going to show you how we are taking clues from the life that exists all around us, and there's a lot of life that exists all around us, and we're using those clues to sort of reconstruct the origins of life to maybe understand how it may have emerged all those years ago. Okay? When I tell people that I work on the origins of life, then oftentimes they will ask me, do I think that there is life in space? And many of you have this question as well. Is there anybody out there? For the older folks, this is actually a rephrasing of a famous line from a Pink Floyd song, is there anybody in there? Which is one of my favorite songs. But this question is so important that the umbrella discipline that origins of life research falls into is actually called astrobiology, which literally means the study of life in stars or space, right? Of course, we don't yet know if there is life in space, but the field is called that. We have actually tried to look for life in space, right? But I'm going to give you some results, okay? So these are some data that my physicist friends have told me. So there are about 1 trillion galaxies in the observable universe, and you have to understand that a lot of the universe is still not observable. So there's 1 trillion galaxies in the observable universe, and these galaxies they have their own solar systems, they have multiple planets in each solar system. So there are about 1 trillion planets. It's almost unimaginable. I cannot imagine how large this number is, right? And then, of course, not all planets are Earth-like. They wouldn't be able to support life, right? Very hot, very cold, wouldn't work for life. But at least we think that from statistical extrapolation, we think there's at least 1 billion trillion Earth-like planets in the universe, which is not a small number. And of course, we cannot forget moons that are associated with many planets. So there are about at least 1 billion moons to choose from, right? So again, if I had to bet in a cosmic casino somewhere, I would go strong on, yes, there is absolutely at least one example of life outside of the Earth, right? Okay. Now, we have, as I said, gone looking for life. So this is the solar system, and we have sent rovers to Mars, for example. We have sent humans to the moon. We'll be sending more humans to the moon very soon. And then we've had these missions, these flyby missions, these orbiters. These are spacecrafts that have gone around these planets trying to image its atmosphere, trying to see if there's life, at least in our solar system. But there is no breaking news here. We still don't have any life that we have detected in the solar system, at least. But the solar system is a tiny fraction of what the universe has to offer, right? So there are these planets. Most planets are actually outside of our solar system, and we call them exoplanets. And there are many exoplanets that are Earth-like, which potentially may harbor life, right? So we should go look for them. But the thing is, and scientists are really excited about exoplanet research now, but the reality is that exoplanets are really far from us. So even the closest exoplanet is 4 light years away, which means you would take 4 years to get there, only if you're light, and we cannot travel faster than light. Realistically, 70,000 years, just for a one-way ride to the closest exoplanet. And then, of course, planets that are farther away, don't even imagine it. So we have to face this harsh reality. Although it might sound like a downer, there's really, we cannot really go looking for life in space. Like Star Trek is not happening anytime soon. And even if we do detect life in space, like this little buddy, if you remember Marvin the Martian, if we detect life in space, we still haven't answered this fundamental question as to how did this life come about? How was this life born wherever we detect it from? So this brings us to a rather core fundamental question in chemistry, and I'm trained as a chemist, which is how do collections of chemicals, non-living matter, dead matter, get together to form something like a living cell? Or as my title says, how did life emerge from scratch, right? Now, of course, life didn't emerge. When life emerged, it was a fairly simple form of life. It was much simpler than the simplest forms of life that we know of. So we think of bacteria, viruses being really simple, but the life that emerged 4 billion years or so ago was way simpler than bacteria. But a cell, if you've ever looked at a cell under an electron microscope, which probably many of you haven't, but a cell is incredibly complex. But at its core, and as a chemist, I like to simplify biology, and sometimes my biology colleagues get angry with that. But a cell, essentially, at its core, is 3 things. So the cell needs genes, right? So genes hold the information, the genetic information of the cell. It tells the cell what to do. And then it also needs enzymes. Enzymes are the workhorses of the cell. They do things in the cell that keeps the cell alive and functioning. And then we need a compartment to put these things together to keep everything together. So those are the 3 things that we need for a cell to work. Okay? So genes, enzymes, and a compartment. And now I'm going to show you some biology here, okay? And most of you would know parts of it. So what are genes made of? DNA, right? So genes are made of a substance called DNA, we all know that. But surprisingly, and it's really curious, that our cells store all our genetic information in DNA, but has no way to read that information. So what it has to do is it has to rewrite that information in the language of RNA, or more specifically messenger RNA or mRNA, which is the stuff in your COVID vaccines. And then the cell reads this RNA message and makes proteins. And proteins, as I just mentioned, they are enzymes. They are things that do things for the cell. Those are the workhorses. And this little workflow is the blueprint that all life uses today, from bacteria to blue whales. So I like to tell people that life uses the same operating system, but just in different form factors. And we are all made of the same stuff, which is, if that's not the most unifying thing, I don't know what is. But regardless of how good this workflow is, there's an issue with this workflow. And if you look at this in detail, you'll realize what that problem is. So I'm showing you here that, it's not working regardless. So I'm showing you here that you need a protein enzyme to copy DNA information to RNA. And I'm telling you that you need the RNA to make proteins. So you need RNA to make proteins, you need proteins to make RNA. What came first, the chicken or the egg, right? It's a problem. And this is what I'm trying to tell my artist collaborator who made all of these, this is what I'm trying to teach him, that there's a problem with this so-called blueprint of life. So when life emerged, it definitely would have emerged without this sort of self-referential, complicated problem. So how can we get around this self-referential paradox? What if there was one entity that could function as both genes and also as enzymes, so we would not even need that messenger connecting these two? So again, we are at Notre Dame, so you can liken this to the Christian concept of the Holy Trinity, right? The Father, the Son, the Holy Spirit, all in one. We need a molecule that can behave like that, all in one. Can we find that molecule? Maybe that molecule holds the key to the origins. And then there are two discoveries in the 19th century that really opened the doors for this sort of thinking. And the first discovery made in the 1910s, when they discovered a new form of life, which we now know as viruses. And the interesting thing about viruses, the ones that they were discovering, is that viruses had genes, of course. But their genes were not made of DNA, like you and me, but were made of RNA, which showed that RNA is capable of functioning as genes. And then a discovery later, in the 1980s, by an assistant professor in Colorado, so where he was trying to study a process where RNA in cells were cut and joined. This process is called splicing. And he was trying to discover the protein-based enzyme that did this function. And accidentally, he found none, but he found that there was an RNA enzyme that did this. So this showed that RNA can function as enzymes. So the fact that now RNA can function both as genes instead of DNA and as enzymes instead of proteins led to the formulation of this now widely accepted model for the origins of life, which is called the RNA world hypothesis, which really paints a picture of an early stage in the Earth's history. Maybe you can date it back to 3.8 billion years or so, where the first forms of life that crawled on the Earth were powered by RNA and not DNA or proteins. So now I'm a chemist, right? So when I think of how we can understand life, how we can reconstruct the origins of life, I see this. And this question of how life emerged from scratch to me becomes, how did RNA-based life emerge from scratch? And now if I have a rough idea of what that form of life would look like, chemists like to mix things and create new things, so maybe we can create this little thing in a test tube in a lab at Notre Dame, which is why I started my lab here last year. Okay, so this is the recipe for creating life in the test tube, okay? Fairly simple. And one thing I should mention here is that I don't want to give you hand-wavy explanations because they will sound like miracles. So I'm going to actually tell you exactly what we do or what needs to be done. And even if that means I have to show you some results from real experiments, that's fine. I trust that you will appreciate that more than hand-wavy explanations, right? Although this is a popular science talk. Okay, so I'm going to tell you exactly what we need to do. So all life uses cells, and cells have a membrane around them, protecting the cell interior from outside, and they're made of substance called lipids, and we'll get to this in a while. So we need to then figure out a way in which you could take these lipids, put them together so that you can get this nice little membrane, a structure, and a compartment. And the second thing that we need to do is because early life was potentially, most possibly, powered by RNA, as I just said, we need to take pieces of RNA or building blocks of RNA and then put them together, find a pathway to put them together so that we can get these large complex RNA molecules that can function as enzymes. And then we need to put these together, right? So this is a model of a primordial cell. We often call it a protocell. And then we need to make this model of a primordial cell grow and then divide and evolve, right? Just like normal biological cells do. And if it does that, then we can say that we've made life in the lab, and I'll show you exactly how it would work. So this is an animation, not real results. This is a dream experiment that someday I hope to do in my lab. But this shows you what would happen if we are able to put this thing together. Okay, so before I start the video, this is what you see on screen. This red little thing is a primordial cell, let's say. It has a membrane containing lipids, as you'll see. And you have these building blocks. So again, these are pieces that you need to put together to make RNA. Okay? So let's see what happens if this thing plays at all. It does. Wonderful. So you see that there is this cell. There is enzymes made of RNA inside of this cell. And what it does is it takes in the building blocks that are swimming inside from outside and using those building blocks to create copies of itself. It's like using bricks to build a wall. And once you do this several times, you have lots of RNAs that are building up inside of this cell, so it grows fat, and then ultimately it sort of distends, and then it will, as you see, break apart and form two daughter cells. This is not exactly, but more or less how cell division works in you and me. But you have to remember that there is no life here. It's all chemicals that we have put together. We've engineered a structure that looks like a cell. But if we have a population of these cells growing and dividing on their own, then it is bound to evolve, right? So if you look at, this is how NASA defines life, you might define life differently. But the scientific sort of working definition of life is, if a chemical system that is self-sustaining can evolve biologically, that is Darwinian evolution we call it, then it is in fact no longer chemistry, it becomes biology. So if we can create these sort of self-replicating primordial cell-like models, and we can make them grow and divide and evolve, then we can actually say that we have created life in the lab. But we haven't yet gotten to this phase, but we are trying to. Okay? But one thing, the reason why I show you all of this is because I think one thing you notice here, in order for all of this to happen, you need enzymes, RNA-based enzymes, that can make more RNA from short pieces. That is the key part. You need RNA enzymes that will stitch together pieces of RNA to make longer, complex RNA. Which is why it's a big challenge to get these enzymes. And the bigger challenge really is that none of these enzymes exist today. So however important these were, 4 billion, 3.5 billion years ago, now we don't have these enzymes anymore. They've gone extinct. So how do we bring back what's extinct? Hollywood did that with dinosaurs. We can do it with molecules. So this is a method that we call in vitro evolution or artificial evolution or test tube evolution, however you want to call it. So how evolution works in biology is that you have a population of organisms, and then a subset of those organisms can survive better than others, they can make babies better than others, and their lineage gets passed on. In case of artificial evolution, we don't care about organisms, we care about molecules. So we take a collection of molecules, RNA molecules, not a small number, 100 trillion different RNA molecules in a test tube. And we challenge this with a particular function that we want these molecules to do. So what is the function we are most interested in? We want these to join pieces of RNA together. Okay? And once we do that, we have a way to select out the ones that can actually perform this function, take them out. So like if you have a room full of kids and you give them Lego pieces and ask them to build a Lego car, and then you take the kids that are successful into a different room and then say, okay, now make a Lego bus and then make a Lego rocket ship, whatever. So you can do this process with molecules. What you cannot do with kids, you can do with molecules, is that you can actually make more of the ones that you select. And then you can do this several rounds, and you can actually, at the end, if you're lucky, get enzymes made of RNA that can make long RNA, right? Okay, and we actually have done this. This is actually a big focus of my lab's research now. But we've identified evolved many such RNA-based enzymes. So enzymes made of RNA, not proteins, that can make long RNA by stitching together shorter pieces. And I hope that you recognize the significance of this, is because if the enzyme itself is made of RNA, and it can take short pieces of RNA and put them together, there's a potential that this little thing can copy itself. And if you have a few molecules of this RNA enzyme, it can very easily copy itself. So you can go from 1 to 2 to 4 to 8 very, very quickly, right? So this is really attractive because we want to create this self-replicating system. But there's, again, a sort of a chicken or egg paradox here. For those of you who are listening to this carefully will ask me, you need this RNA enzyme to make more RNA enzyme. How did the first one come? Where did the first one come? And that's obviously a serious problem. This has been a problem in the field for a long time. And we've found somewhat of a solution accidentally, which is very exciting, is that, okay, if you put these pieces of RNA without any enzyme, nothing really happens. They don't come together. You need this enzyme. So we found that if you incorporated amino acids, which are the building blocks of proteins, and we have evidence, plenty of evidence actually, that amino acids would have been present on the early Earth 4 billion years ago. So if you incorporate amino acids into this mixture, it's seen that these pieces of RNA can actually come together and react with each other very slowly, but surely they can come together, and they can create these long, complex molecules that can themselves start functioning as enzymes, right? So that explains how the first enzymes could have emerged on the Earth. And the solution is nothing fancy, no engineering. It's pretty much amino acids. It's just very, very easy to happen on the early Earth. So again, as I said, we have created many such, some of my students are here, they've created many such enzymes that can take pieces of RNA together and make longer pieces. So I hope I was able to somewhat convince you that we have some advances in this area of research. But of course, not the whole thing. But the big goal is now to then create a lipid membrane and create a model of a cell. Like, that's the exciting part. Can we then put these enzymes in these cell-like compartments and see if they function inside of them? And that's the first step to creating artificial cellular life in the lab. Okay, so I'll spend the rest of my time talking about how we can build that cell. Okay, so our cells have membranes made of lipids, and these lipids are called phospholipids. These are quite complex, actually, chemically. A lot of different steps go into making these, a lot of different enzymes. So it's hardly possible that early life started with phospholipids. So it must have started with something that was simpler. And I always look for something that is similar but simpler. And fatty acids, of course, you don't know what fatty acids are. Fatty acids they look like phospholipids, but they're not. They're simpler than phospholipids. And there's several interesting aspects of fatty acids. I'm just mentioning a few of them. So essentially, we think that the chemical processes that were going on in the early Earth 4 billion years ago were sufficient to make these fatty acids at high temperatures, for example. And something that really is still amazing to me is that we have these meteorites that have crashed from space onto the Earth. This is an important one in Australia. This is called the Murchison meteorite. And when this crashed, scientists went there, collected samples, and then they analyzed these samples, and they found fatty acids in meteorites. So all of this tells you that we don't need talented graduate students at Notre Dame or wherever else to make these molecules. Nature makes them on its own, right? So these molecules would have been present before life emerged on the Earth. The next piece of data that's really exciting is that if you take, and you can buy these, essentially soaps are salts of fatty acids, as you all know, right? So you can take fatty acids, put some RNA, very easy to get, mix them in a tube, leave them overnight in a tumbler that goes round and round. And if you come back and look under a microscope, you will see something like this. These are called spherical vesicles. So these are spherical things where you have the RNA in green because we put a fluorescent dye on there, and the fatty acid membrane is in red also because we added a dye to that. So these kind of look like cells to me, right? So the big test for this would be if we can put some of our enzymes that we have evolved in a test tube that put together these RNA pieces together inside of a primordial cell and see if it works. If it doesn't, then that's a big setback, because then that's the first step. This is a very simple system to mimic a living cell. So we did this experiment. So what we did was, again, an experiment that any of you can do, which is just get some fatty acids. You can buy it from a company. We put the RNA enzyme that we evolved that stitches together pieces of RNA. We also put those pieces that we want stitched and mixed it, right? And left it for some time. And then when we broke open this cell, we wanted to, because this was the first time that we were testing this, and we didn't know if this would work or not. So I don't want to show you the data here, but what we found was that this enzyme that we had entrapped within this primordial cell was able to join these two pieces of RNA to make a brand new long RNA, which means that these ancient enzymes that we are bringing back to life in the lab actually are able to function in these lipid models, lipid membranous models of primordial cells. So this is perhaps the first time an RNA enzyme has made a new RNA inside of a primordial cell in 4 billion years. So this is a really exciting sort of stepping stone for us, and we are using this to go forward and really create something like in the video, something that can self-replicate. So this is a zoomed out view of what it takes to get to life. I don't want to read this slide out. I just want to show you we have to start with a planet that we call habitable, that can support life. It needs to have liquid water, most probably. It needs to have moderate temperature, pressure, and so on. But what this slide really says is that once we have a habitable planet, all of these things actually happen through simple forces of physics and chemistry. You don't need to invoke a lot of complexity to this. And once we get to something called LUCA, which is the common ancestor of all of us, all of life, I mean, once you get something that is evolving on its own, then Darwin has taught us how evolution works. So then evolution takes care of all the complexity and gives us humans. And sometimes when I look at this, I think to myself that humans are the only forms of life, as far as I can tell, that can even ponder our own origins, that can ponder the origins of the cosmos. So, in a poetic way, life is the universe's way of looking at itself, perhaps. That's something to think about. So we're almost at the end. I just want to end with different aspects of origins of life. This is a fascinating area of science which very few people really know what's going on, which is why everything that we do is new and exciting. But the origins of life is also one of the most, if not the most, interdisciplinary field of research there is. It allows me to talk to academics at Notre Dame and outside in disciplines that I have no idea what's going on, right? Like planetary sciences and geology, philosophy, theology, especially at Notre Dame. And it also allows me to talk to non-academics, football fans like yourselves. And that's a really, really interesting, exciting approach to doing things. And this is one example of how I'm trying to figure out or work out new ways to talk to the non-specialist. And this is, again, in the form of a graphic novel that we're writing. I'm writing with my artist friend over there. This is the cartoon comic strips that you saw in my talk are from this graphic novel. These little posters are also from that graphic novel. These are pages from there. These are actually from an exhibition that we held at Notre Dame in the summer of this year. We launched a couple of books here. I have some of them with me if you want to take a look. Honestly, the whole goal here is that none of us were there when life was born almost 4 billion years ago, but we are trying to bring that moment back using a scientist's precision and an artist's imagination. We need a lot of imagination. Okay, so in the grand scheme of things, we still have so much to learn. We don't know much, but we know some. I think, especially for me, I think it's a great feeling to be constantly challenged and humbled by these big, profound questions on a daily basis. It's not a bad thing. I think it's a privilege. In the grand scheme of things, I hope that I was able to convince you somewhat that we are closer, much, much closer to making life from scratch in the lab than you might have thought of, right? So we are quite close, which is why we are not there yet, but we are getting there, right? And so I am actually, honestly, with some of my students here, I am really excited about the long-term prospects of origins of life research and creating life in the lab. And what I'm also excited about in the short term is that I'm going to attend my first football game ever tomorrow, right? So hopefully I'll meet some of you again there in the stadium. Thank you. That's all. I can take questions if there are any.
We have time for, like you said, a few questions. If you are able to stand up, that would be great so we can get you on camera, and I will pass a mic around for your questions. If you're not able to stand up, that's fine too. Don't worry about it.
I'm able to stand up. I don't know if I'm going to be on camera. Hello? Okay. I'm at the football fan level. I'm not going to tell you what I majored in, but it was a long time ago. But can you just repeat because it seemed like it was the most startling part of what you're doing. Is it true that you made RNA replicate itself or join to make longer RNA? Is that true, what I just said, or?
So what you're saying is that, okay, is it true that I've made RNA that replicates itself, and is it true if I've made RNA that can join RNA pieces together? Yes, so it is true that we have made many RNAs that can join different pieces of RNA together, like I showed you right about, let's see, here. But truly self-replicating molecules where you have one RNA that creates a copy, that creates another copy, that is still not done. So that is something that there is a potential. So once you have an RNA molecule that can create long RNA molecules from shorter pieces, there is a possibility, technically speaking, that you can get to a self-replicating system, but it's just technically challenging. It's theoretically very, very possible. We're trying to get there.
Okay, so the part where the smaller RNA get together and make a bigger RNA, that's a chemical reaction, but to make it replicate itself, that would be the biology. Is that where the life part comes
very
in.
interesting. So the question is, when is it life, right? When is it biology, right? So yes, so when an RNA molecule creates a copy of itself, I would argue that is still a chemical step. When it becomes biology is when it, so that's really getting into technical weeds. So basically, when something replicates with variation, right? So like Darwinian evolution is replication with variation, slight variation. So when something replicates with variation, and then natural selection can act on it and select certain molecules out of the other, then it's life. So what I think would happen is when we call something life is this slide that I showed you earlier. Let's get, oops, something bad happened. Let's see, I'm gonna go back and see if this works. Okay, right here. Yeah, this is the one. This is when you have these little models of cells with RNA inside, and then they self-replicate. So you have 2 from 1 and then 4 from 2. And when this happens, you can call this life, because now you have a population of these sort of cell-like systems that are replicating, copying, growing and dividing, and that's when evolution can actually act on it. So our natural selection can act on it. So again, I'm happy to explain this further. This is hard.
Thank you.
Yes.
Are you the artist for the cartoon work?
Oh God, no. No, I'm the scientist.
Okay, well, it's a fascinating way to tell a complex scientific story, and I think they're fun, and they look like R. Crumb.
What was that?
Have you ever heard of R. Crumb?
I have not.
He's an artist.
Oh, okay.
Similar style.
Oh, okay.
I also like the term binge thinking.
I found it on a poster somewhere on campus. I used it. But the artist that I'm collaborating with is a biology PhD dropout. So he understands biology quite a bit, which is really helpful here. Yes.
Yeah. Two things that you went through quickly, and I just want you to kind of clarify. One is when the RNA enzyme is being created, the one which then can fabricate more. That is created, can you go over the details of how that's created again? And then the second question is, just from what you explained to him, when you are creating, when you're bringing many of these small pieces together through the enzyme, creating longer RNA, are they, at this point, are they not the same RNA? There are many different long strands of RNA, there are many different types. You're not doing the same type.
Okay, so the first question was, where did we get this RNA from? Yeah. And that is basically artificial evolution, like I showed you earlier. So we were basically evolving RNA enzymes that can perform this joining reaction between two RNAs. And this was done by this process essentially. So we had a library, a collection of many RNA molecules, and we evolved those populations, and we ultimately ended up with several RNA enzymes that could join pieces of RNA together, and that led to this particular molecule that you see here.
So this came out of a sort of Darwinian or quasi?
Yes, absolutely. Yes.
Okay, but it's, you didn't manipulate it, you just let it happen.
Exactly. So that's the beauty of, so again, so we were talking earlier, and you're an engineer, and people that do this are referred to sometimes as Darwinian engineers. So the difference between a Darwinian engineer and an actual engineer is an engineer actually intentionally designs something from piece by piece. But a Darwinian engineer uses combinatorics, takes a population of stuff and pushes it through some sort of filter and sees what it gets at the end. And it's leveraging natural selection and Darwinian evolution as its main driving force. It's not intentional.
You're using a filter, essentially.
Yes, exactly.
And recursively going through it.
There is no intelligence. There's just, the only thing we are doing is we have this collection, we have devised a way to pull out those that can perform this function and make more of it. That is the only human intervention that we are doing. The rest is just forces of nature, really.
And your supposition is this happened in nature.
This does happen in nature. I mean, we have so DNA evolves. That is how evolution happens. Evolution happens in the level of our genes, right? So this is actually happening, not exactly the way I'm showing you in test tubes, but it's happening inside of our bodies. We are the test tubes for evolution. So this is actually happening. It's just that we are reconstructing evolution in a test tube outside of a cell. That's basically it. And again, this idea is not new. This was discovered decades ago, this technique. But in the in the broader context, it is new because now we have a way to reconstruct or bring back these lost enzymes of the RNA world. Yes.
Thank you for the very nice talk. Just a question on the protocell. Did you use UV radiation at all to do any of the chemical modifications? Was that involved?
That's a good question. UV is, so short answer is there was no UV involved in this experiment or the experiments I showed you, but as you would know that the Earth didn't have free oxygen for a long time, and then which means we didn't have ozone. The ozone layer was not there, which means that high energy UV percolated through oceans. And so UV definitely played a big role in deciding what chemicals survived. And that would then go on to give all of these processes. So basically from RNA. So RNA is made of building blocks called nucleotides, and that's details. But some of these might not have survived. Maybe we had many different building blocks and only 4 survived. And maybe UV had a role to play in that. Actually, I know someone who works on this area. Very interesting, yeah.
Last question for today.
Hey.
How is it planned to make the RNA split once it has combined?
So I think you're talking about how RNA 1 becomes 2, is what you're saying? Right. That's the heart of replication. How does something replicate, right? So to replicate, it needs to, so you have an RNA molecule, right? And you want to make another copy of it, right? So all you have to do is take the pieces that make that molecule, you add them up, right? So you can make a copy. So what this does is it actually, so you have the RNA molecule, it takes the build, so okay, let's think of this way. Like you have a string, like a necklace, right? It's made of pearls or beads, right? So if you can get the same beads, and if you put them in the same order, you can create an exact copy of the necklace that you have. So RNA is like a necklace. It's made of 4 different kinds of beads. So if you know the exact order in which those beads are put in the necklace, and you buy those beads and a string, you can make an exact copy. So this is exactly how RNA works. RNA takes these building blocks, puts them on itself, and then creates a copy.
All right, thank you very much.
Thank you. Thank you for coming. Really appreciate it.